Good afternoon, everyone. It's a pleasure to be here presenting our recent data and our mission for liver disease patients. Let me know if you don't hear me well. I think it's a good setup, so it should be fine. So we are a company located in Zurich, Switzerland, and focus on liver disease. The reason why we focus on liver disease not only is a very hot topic, growing commercial opportunity, as you may know, but also because the unmet medical need is actually huge in the space. And I think there's nothing better than a little scary slide to exemplify the unmet medical need. What you see here is actually the mortality rate in the UK over the past 40 years. And the good news is that disease such as diabetes and cancer can be treated. Actually, the mortality rates are decreasing, but the story is completely different when we talk to liver disease. There is a huge 500% mortality rate increase that we've seen over the past 40 years. In the UK was mainly driven by alcoholic hepatitis back in the days, but things are changing currently. The NASH, so the fatty liver disease, is playing a major role into the growing of this mortality. And obviously liver disease is a very large space, and cirrhosis is the end stage of many of these liver disease. Cirrhosis kills 2 million people every year, and in the US as well as in the rest of the world, the mortality rate is currently growing at epidemic rate, and obviously, you know, the medical expenditure as well. Right, so again, liver disease has many different causes actually, so obesity or viral infections such as hepatitis, alcoholism, we've seen it. So all of these drive actually the liver disease indications. And liver disease is also a space which is well covered in terms of very good medicines. So blockbusters in hepatitis as well as promising drugs in NASH. But all of these drugs are preventive treatment. They're all trying to prevent cirrhosis, but the patients which has cirrhosis, and especially when it decompensates from cirrhosis, can today only count on a liver transplantation. It's today the only cure, right? And in the acute care of decompensation of cirrhosis, there is nothing to help these patients. So that's exactly where we position our first and principal product, which is VS01, in patients with decompensated cirrhosis, and in what you see on the right-hand side, ACLF, which is a very interesting indication called acute on chronic liver failure. It's interesting because it's a comorbidity indication, and you'll see that VS01 can treat simultaneously these different organ dysfunction. It's also interesting for us because it's a rare disease, and we have the orphan drug designation at the FDA and soon at the EMA to streamline our clinical development pathway. So you see we have 2 indications there in liver disease, and a third one which is interesting as well in pediatric indications. So what's the problem in this patient with liver disease? When they decompensate, they can no longer metabolize the toxins that your body generates, right? So the major role of the liver, which is to clean your blood, is not efficient anymore. And to treat this basically intoxication, we can today only rely on chronic therapies, or in a very last resort on when the escalation of care is happening in hemodialysis in the ICU. However, these hemodialysis techniques are today not functioning. They have been several clinical studies and nothing has been proven to be ameliorating the survival of these patients. So there is a huge medical gap that needs to be filled between these chronic drug therapies and the last resort ICU treatment for these patients with severe liver decompensations. What is very important, that's what you see on the right-hand side here, is to treat this patient early. What you see here is basically the hepatic function, and you see that after the injury, the decompensation leads very rapidly to dramatic mortality rate. So the speed at which patients decompensate is extremely impressive, and you have approximately 1 week to bring a treatment to this patient to make a difference to these patients. Otherwise, the fatalities can be dramatic. And that's exactly in this golden window of approximately 1 week that we position our product VSO1. So it's going to be an acute product, a hospital product, to help this patient fight and survive this decompensation event, to eventually go back to the cirrhotic stage they had before and be stabilized, or to prolong relatively their survival in order to get a liver transplant eventually. So what we have at hand and what is the product is a liquid solution that contains microscavengers, which once injected in the peritoneal cavity of the patient will capture the excess of toxin which is circulating in the blood. And you will think that that seems very invasive. However, these patients with decompensated cirrhosis arrive to the hospital with ascites, which means that they have fluid accumulated into the abdominal cavity. And the very first medical gesture which is done in this patient as standard of care is to place a catheter and to drain this ascitic fluid out. And we are the first company to use this entry route to treat this patient with decompensated liver disease. So we'll use this catheter, which is there as a standard of care, to inject our fluid solution. It's approximately 2 to 3 liters that we inject into this space. But remember, this space is usually filled with ascites, so they have up to 6 to 10 liters of ascites. So there is enough space for accommodating our 2 to 3 liters of fluid. Our fluid contains microscavengers, which are based on a liposome technology that we designed proprietary. And these microscavengers will sit there for 2 hours and have the time to capture the toxins which are in excess in the body of these patients. These microscavengers are not absorbed systematically. They're really staying into this abdominal cavity, and after 2 to 3 hours we simply remove them, drain them out the same way that we drained the ascitic fluid before. So it really fit the patient, as it's very similar to the standard of care which is today administered in this patient in the hospital. The mechanism of action of our product is twofold. First of all, we can efficiently and selectively capture ammonia, which is the major toxin in these patients with decompensated liver disease. We do so because inside of our vesicle we have an acidic medium, and this acidic medium will protonate all the molecules of ammonia which is penetrating inside the vesicles. Once it's protonated, the molecule will not be able to escape the liposomes anymore. Therefore, we create a sink for this ammonia, a sink for the toxin, and continuously drag the ammonia out of the blood circulation inside the vesicles. So we can really concentrate ammonia there, and that's how it is so effective at depleting ammonia from the blood circulation. So this is one part of the mechanism of action that's very important for what we call hepatic encephalopathy, because ammonia is a very strong neurotoxin. And on the other side, we also remove other toxin by passive diffusion inside the supporting fluid. And we've shown that we can remove toxin which are related to kidney and liver toxicity as well. So we can really treat the patient on a global level by supporting the brain, the kidney, and the liver. And that fits very well with this indication, comorbidity indication of acute and chronic liver failure. Obviously, we've done a lot of work to optimize the technology. They are liposomes, which are actually based on components which are either endogenous or have been approved in other pharmaceutical products. And that was very important to guarantee the safety. So we know that each of the components of our formulation are safe. And we also worked a lot on the efficacy and stability. The membrane should be sufficiently permeable to let the molecules of ammonia penetrate, but also sufficiently impermeable to avoid the leakage of these molecules or of the acidic buffer inside. Right now, the manufacturing setup is scaled up, is GMP manufactured. and we have a long-term shelf life of the product already, and these data are being generated as we speak. In terms of product, we'll package the solution in a multi-chamber bag, and that will be reconstituted just before the administration of the patient, just in order to make sure of the stability, of the sterility. So all of these solutions are autoclaved, so it's a very upscalable process, and also to make sure of the efficacy of the product before the injection into the patient. In terms of data, I'll show you just 3 slides today. What we've shown, and again this is focused on ammonia because it's our biomarker, but we can remove other toxins which are implicated in the comorbidity of this patient. What we've shown first is that we can concentrate ammonia in this peritoneal cavity, so we can really enhance the clearance of ammonia, and we've shown that the concentration of ammonia is up to 20 times higher in the peritoneal cavity than it is in the blood or in the plasma. So that really validates the mechanism of action, and that concentration of ammonia in the peritoneal cavity translated into a significant reduction in the plasmatic concentration after just 3 sessions of the administration of the product, and that reduction in the plasmatic circulation translated itself into a significant reduction in brain edema, which is actually the readout for hepatic encephalopathy. So with this data, we validated the mechanism of action, and we showed that it's that it's efficacious in a clinical relevant endpoint, which is the brain edema in bile duct cirrhotic rats. Then what we showed as well is that we can remove these other toxins which are related to the kidney and the liver decompensation as well. As I mentioned, we capture ammonia inside the liposome, but we've shown that we can also remove other toxins which are hepatotoxin or uremic toxins. Up to 70% of the toxin related to this kidney and liver decompensation are being removed by our product. And then what we're generating now is to understand what are the toxins that we can capture within the membrane. We know that it works in other products, and it will also work in the peritoneal setup with our liposomes. So that will be interesting to understand what kind of protein bound toxin we can remove in these patients. In terms of safety, we've proven the safety in rats, we've proven the safety in pigs and in mini pigs as well throughout our preclinical development. And we are right now conducting our first in-human clinical trial. So it's a phase 1b clinical trial in decompensated cirrhotic patients. It's set up in 2 different parts. So part A is a single ascending dose. In our case, the ascending dose is actually ascending volume. We inject first 1 liter, here 2 liters, and here 3 liters into the abdominal cavity of the patients. And the part B is a multiple administration, so 5 days in a row we'll treat these patients. Cohort 1 is currently completed, and there is no safety issue, there is no adverse event, no study discontinuation. The patients feel really good after the treatment, and we are currently enrolling the second cohort. So as you see, phase 1b will be completed within the course of this year, and we will start conducting a phase 2a in 2 different indications. So the first indication is decompensation of cirrhosis, and the second indication is this acute and chronic liver failure, which is the rare disease of interest. Because it's a rare disease, we can get a marketing approval quite rapidly, and after this phase 2A trial will be just 1 study away from the marketing approval. Now comes the third indication. It's a pediatric indication called urea cycle disorders, and in this indication patients are born with an enzymatic defect, and they accumulate ammonia as soon as they start drinking the milk of the mother, right? And therefore it's also important in this population of patients to remove ammonia as fast and as efficiently as possible. It's an interesting indication because we can get in the US the priority review voucher, which is quite valuable for our company. Of course, as I mentioned, the field of liver disease is quite interesting in terms of transactions and deals as well. And you see that it's a field being very active recently in transaction, in NASH obviously, but also in hepatic encephalopathy and urea cycle disorders as well. And these are just a few examples of companies interested in the field. And we have the right team to move the project forward. Miriam is the co-founder of the company, and she has more than 10 years business experience in the space, developing a clinical trial and running clinical trial with Celerion and MDS Pharma. Sophie Biggener is a physician, pediatrician, and liver transplant surgeon as well, and has a long career in the US in different pharmaceutical companies. And Peter is our chairman, and he is a senior pharma executive working at Baxter mainly over the last few years. And to finish today, we have a very interesting product. It's a low-risk product, first of all, because the safety is de-risked. All the constituents have been approved and have been used in other products. And in terms of mechanism of action, what is interesting in our product is that it's purely physical, it's purely mechanical. So we don't rely on a specific binding, on a specific interaction with one molecule. We rely on passive diffusion of toxins, which we know happens the same in animal that it actually happens in human. We have a lean clinical development pathway thanks to our orphan drug focus, and obviously higher commercial opportunity and business upside with the different indications that we have. So with that, I will thank you for your attention, and I'm very pleased to answer your questions.